Hello friends, Ace Botany next episode. In this episode, we'll be discussing a new chapter, Cell, the Basic Unit of Life. A theoretical chapter, but for exam point of view, it is important for both theory and NEET exam. So for convenience, I will share the screen. Now what else we have to discuss remaining chapters? After Cell, the Basic Unit of Life from first year, biology classification is remaining. So by which first year topic will be over. Second year, ecology, we started. Ecology 2 chapters are available, by which all chapters in second year will be over. Connected with the botany, that differs in different places. After that, we'll be discussing each chapter or difficult portions of each chapter in small segments. So for you, it will be easy for reference. So meanwhile, all the notes will be there in YouTube for all botany chapters. Chapters may vary according to institutes. Okay, so we can proceed. So I will share the screen, and then it will be easy for you. I think a screen came, so we can proceed. So what all things we'll be discussing? So a new unit itself. So unit 3, cell structure and function. And here what we are to discuss is chapter 8, 9, and 10, of which chapter 10 is already over cell cycle and cell division. Chapter 9 will be taken by zoology teachers. Now chapter 8 will be discussing cell, the unit of life. I think first page is clear to all of you. So we can proceed to second page. See, this page is purely NCERT. So you may wondering why I put this NCERT page, because specific reasons are there. First, students will not read at all these areas, these pages from NCERT. So few sentences I will show you how it can come for the question. So you can find first red bracket here. What they mentioned is, it is the cell theory that emphasizes the unity underlying this diversity of forms. Cell theory. That is cellular organization of all life forms. So that can be a question. It is the dash theory, cell theory. Another one here. Cell theory also created a sense of mystery around living phenomenon. That is physiological and behavioral process. That is second one. Now, third one here. This physicochemical approach in study and understanding living organism is called reductionist biology. So that approach, reductionist biology, what it means. So this way question can be put. For that purpose only, I put this page from NCERT to show, because otherwise people will neglect these pages from NCERT. Don't do it. Now, next page, you will be familiar, famous Indian scientist Ramachandran. It's a usual question. His famous work is concerned with amino acid and proteins, and he created a system called Ramachandran plot. He worked together with the famous scientist Linus Pauling. So, an authority in chemistry, Linus Pauling, etc. So, the Ramachandran plot, G. N. Ramachandran, famous Indian scientist, connected with the amino acid and proteins. Now, we will be proceeding to next page. Here the chapter begins. Chapter 8, Cell, the Unit of Life, Part 1. Topics, Introduction, what is a cell, cell theory and overview of cell, prokaryotic cell, starting. Then remaining topic we'll be discussing in next episode. So introduction. See, only two types of things in this world, what NCERT says. One is living, one is non-living. Living is animate and non-living is inanimate. What makes the difference? Only due to basic unit of life, and that is called cell in all living organism. That is how it differs. That's what NCERT says in page number 87. Now, what is a cell? Cell is, four terms I written there: structural, functional, basic, fundamental unit of life. That is cell. And when we describe life, all these terms will be coming. Structural, functional, basic, fundamental unit of life, that is cell. Now, below that, we can find four sets of important words. One by one, we can discuss. This way, you may not get in textbook. It will be like sentence, but I put all together for your convenience. Unicellular, multicellular, acellular. Unicellular we know, multicellular we know. What about acellular? Explanation given below. Acellular generally means having no cell, a prefix, acellular. Example is virus. Well, when we come to bacteria, it is unicellular. When we come to animals and plants, it is multicellular. So that three times clear, I think. Now comes prokaryote, eukaryote, mesokaryote, eukaryote. Prokaryote, you are familiar? Bacteria, eukaryote, higher plants and animals. Now what about mesokaryote? Mesokaryote, meso means in between. That's the word meaning meso, meta, all this. So mesokaryote means single-celled eukaryote in between prokaryotes and higher eukaryotes. Typical example is dinoflagellate. Here we can find, I put dinofla, after that no space, that is why I put dot dot dot. Next page when I write it, it will not be clear for you. So mesokaryote example is dinoflagellate. Eukaryote means viruses. Now third set, cytoplasm, protoplasm, protoplasm. What is protoplasm? Cytoplasm plus nucleus. What is cytoplasm? Protoplasm without nucleus. And what is protoplast? Protoplast is normally referring to plant cells without cell wall. Now last four times, undifferentiated, differentiated, dedifferentiated, redifferentiated. Here it comes, anatomy it comes, plant growth and development it comes. Undifferentiated, not able to do any function like meristem. Differentiate, they will lose the power to divide and will be functional, parenchyma, collenchyma, etc. Dedifferentiated means some of the differentiated cells may regain the power to divide, maybe connected to the secondary thickening. And finally, redifferentiated, that is, dedifferentiated cells will again lose the power to divide. That is, last part of secondary thickening. I think these four sets clear. One or two questions we'll be getting usually for theory and entrance. We can proceed to next page. Now, scientists we are going to discuss. Three famous scientists, 2-3 will be coming further. First is Robert Hooke. Second is Anton von Leeuwenhoek. Third is Robert Brown. Now, their contribution I written below, so it will be convenient for you. That is why I told you initially I'll put the notes like this. Then after completion of notes, we'll be discussing part by part. So 1665, Robert Hooke, famous book, Micrographia, and the term cell. That is, originally he worked in slice of cork. Cork is dead. And what he observed was cell wall, like honeycomb. That is 1665, Robert Hooke. Now, Anton von Leeuwenhoek, 1674, observed living cell, bacteria, protozoa, spermatozoa, based his own made microscope. And finally, in 1831, nucleus by Robert Brown in orchid roots. I think these three famous scientists clear. One question we may get usually, even match the following. Now comes another famous theory of this chapter, cell theory. So what is cell theory? Who proposed it? Who modified it? Cell theory tried to explain importance of cell and how it is linked with the life. It is connected with the two famous scientists, namely Matthias Claren and Theodor Schwann. I think that part is clear. What is the importance of it? Who are the scientists involved? Next page. There also you can find two famous scientists, Matthias Claren and Theodor Schwann. Now, regarding details, 1838, German botanist Claren analyzed large number of plants are composed of different kinds of cells which form the tissue of the plants. It's the main work of Claren. Now, Theodor Schwann, 1839, British zoologist, studied different types of animal cells. And he reported there is a thin outer layer, now it is called plasma membrane. Similarly, plant cells have cell wall unique to plant cell wall. It is unique. These are the main contribution. Then their combined one. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cell. That is what NCERT in page number 88 given. I think page is clear. So everything clear in such a manner I put there. So it will be easy for you. Even those who are watching it for observing it for first time, it will be clear. Now we can proceed to next page. Okay. Claren and Schwann together formulated cell theory. That is what NCERT says in page number 88. But one deficiency was there for their theory. Deficiency was they are not able to explain how new cells were formed. How new cells were formed or are formed. So answer, it is given by Rudolf Virchow in 1855 through a famous statement, homine cellula e cellulae, meaning a cell rises from pre-existing cell. He modified the hypothesis of Claren and Schwann to give the cell theory a final shape. And cell theory as understood today, as NCERT page number 88 says, all living organisms are composed of cells and products of cells. Second one, all cells arise from pre-existing cell. Some modification also can be there in connection with these two statements. Now we are entering into cell gradually. So first an overview of cell. So when we come to an overview of cell, for a typical plant cell, example is onion peel. For a typical animal cell, that is human cheek cell inner side. Plant cells normally got cell wall and cell membrane, while animal cell has got only cell membrane. That idea clear, I think. For a typical eukaryote, initially we are trying to explain with a typical eukaryotic cell. There will be a nucleus and cell organ. So cell organs, membrane-based cell organ, it is in typical eukaryote. Now I'm taking a slight deviation. That is why I put the star there and bracket there, slightly going to some common aspects. So eukaryote up to this one, then we'll be continuing later. Next page. Now bracket, cytoplasm is C. Common aspects, both the prokaryote and eukaryote. And next sentence is a famous statement from NCERT, cytoplasm is the main arena of cellular activity. Ribosome is seen in both prokaryote and eukaryote. Remember, ribosome is an membranous cell organ seen in prokaryote also, mitochondria also, plastid also. And ribosome is one of the smallest structure. NCERT page number 88, 89. Now we are continuing eukaryotic organelles here. Here onward, that star is came back. So this is all applicable to eukaryote. In between, some common aspects and prokaryote we discussed. Now chloroplast mitochondria seen in eukaryote. Now animal side, there is a structure called centriole. That is again having no membrane. So ribosome, no membrane. Centriole, no membrane. Nucleolus, no membrane. So usual question, amembranous structures. Odd one. Now cells differs greatly in size, shape, activities. That must be, then only functions can be performed. Mycoplasma is supposed to be a smallest living cell. Smallest living cell, only 0.3 μm in length. While bacteria could be 3 to 5 μm. Largest single cell is ostrich egg, having a weight of 1.4 to 2.3 kilograms, almost 15 centimeter in diameter. Its volume-wise, for a comparison, approximately about 25 chicken egg equivalent. Idea clear, I think. And when we come to multicellular organism, again, some size, length, etc. given in NCERT book, human RBC, about 7 μm in diameter. Nerve cells are some of the longest cell. Single largest cell, I told you it is ostrich egg. Cells also vary greatly in their shape. That is mainly connected with the functions and areas. And the shapes may include, diagram given there, like disc, polygonal, columnar, cuboid, thread, branched, even irregular. This diagram famous, NCERT-based question, match the following. Now we can proceed to next page. So now here we are entering into prokaryotic cell. So prokaryotic cell, which all organisms will be there as prokaryote? Here you can find, includes bacteria, blue-green algae, mycoplasma, PPL, etc. And after that you can find some description. They are generally here. Continuation here, smaller in size and multiply much faster. Since size is smaller, they can multiply much faster than the eukaryotic cells. They may vary greatly in shape and size. But next is a question, exhibit. Here you can find similar basic cellular organization. Similar basic cellular organization. Shape, etc. may change, but basic cellular organization is similar in all prokaryotes. And this is a famous diagram where you can find a statement given here. Sometimes it will cause some confusion because in figure, you can find bacterial size is given 1 to 2 μm in NCERT. But in statement, same NCERT given, it is 3 to 5 μm. So, which is correct. So, what we can say is, best take a combined value, say like a 1 to 5 μm. So, both values we can include. NCERT given 2 values for same size, etc. Same structure. So, 1 to 5, then it will be okay. Otherwise, confusing. I think idea you got. Now, we can proceed to next page. So when we come to next page, we are slightly going to prokaryote detail. We'll be continuing next episode. So most prokaryotes normally have a cell wall surrounding the cell membrane. So cell membrane is inside. Cytoplasm is present. But remember, no well-defined nucleus. Naked genetic material. Genomic DNA seed. It is single. And that next term I put in inverted comma because we can call it as chromosome in inverted comma or circular DNA. Again, inverted comma. In addition to genomic DNA, many bacteria have small circular DNA outside the genomic DNA. Have small circular DNA is a question outside the genomic DNA. This small DNA or smaller DNA are called plasmids. Important term, plasmids. Plasmid DNA confers certain unique phenotypic properties to such bacteria. Example is resistance to antibiotic. Similarly, this plasmid DNA is used to monitor bacterial transformation with the foreign DNA. No other cell organ, but ribosome is there. But remember, ribosome is membraneous, seen in both the prokaryote and eukaryote, 70s, 80s, etc. We'll be studying later. I think idea clear. Now, after that, 5 theory question, 5 NEET model question with the key. Next page, you can find NEET model question with the key. So we can just rewind and come back. So it'll be easy for you. So today we started a new chapter, that is cell, the basic unit of life. A theoretical chapter important for exam. Under unit 3 of NCERT, 3 chapters of which cell cycle is already over. Unit of life we are proceeding. Biomolecule will be taken by zoology teachers. And I shown first initially NCERT pages, 2 pages I shown, and the bracketed one are important. So don't discard it. For that purpose only, I shown these 2 pages. Now Ramachandran plot, protein amino acid link, etc. Then next we entered into topic, cell, the basic unit of life, introduction, 2 types of events or substances in this world, living, non-living, animate, inanimate. The difference is only cell. Now what is a cell? 4 functional aspects given, structural, functional, basic, fundamental, then 4 sets of words. Each set is important and defined it. After that, we entered into next page where 3 photographs, famous scientists, Robert Hooke and John von Leeuwenhoek and Robert Brown, their work and their contribution in science. Then cell theory by 2 famous scientists, Scleden and Schwann, their work, what is the contribution, who worked with the plants, who worked with animal, who described about plant cell, that is cell wall, etc. Then Schwann proposed certain hypotheses, that is bodies of animals and plants are composed of cells and products of cell. All NCERT. Then we continued with this. Scleden and Schwann together formulated cell theory, NCERT page number 88, but one deficiency was there. This theory not able to explain how new cells were formed. Not the place how new cells were formed, how it is formed. Answer given by Rudolf Virchow in 1855 by a famous statement, hominis celluli cellulae. Cell arises from pre-existing cell lineage. Then he modified hypothesis of Scleden and Schwann to give cell theory a final shape. So 2 famous combined statements. Some books, one may be there. This is the essence, this 2 NCERT. Then we entered into an overview of cell, plant side, onion peel, animal side, inner cheek cell. Plant cell got cell wall and cell membrane. Animal cell has got only cell membrane. Then typical eukaryotic cell, there will be nucleus and cell organ. Then we took a deviation. Here you can find cytoplasm is seen both prokaryotes and eukaryote. Cytoplasm is the main arena of cellular activity. Ribosome is seen in both prokaryote and eukaryote. And remember, it is a membranous, one of the smallest structure. NCERT again, page number 88, 89. Then we came back into eukaryote. Cell organs like chloroplast, mitochondria seen. Animals have centriole, no membrane, a membranous, so a membranous 3 structures. Centriole is there, ribosome is there, nucleolus is there. Then after that, cell differs greatly in size, shape, activities. Then mycoplasma, smallest living cell, 0.3 μm. Bacteria 3 to 5, 2 values are there in NCERT itself. Largest single cell ostrich egg. Its weight, its diameter, its volume, etc. Then diagram, match the following. Again, we came to human RBC, 7 μm diameter. Nerve cells will be longest and some branching will be there. And cells also vary greatly in their shape due to function in the area where they're located. Match the following. Then, typical example, mesophyll cells connect with the photosynthesis. So it is round and oval. Tracheate, conduct elongated this way. Then we entered into prokaryotic cell with an introduction. Here, figure bacterial size is 1 to 2 μm. Here you can find. But in statement, previously we studied 3 to 5 μm. So 2 values given, so better, 1 to 5 range. Now, prokaryotic cell, which all organism comes like bacteria, Bluegreen algae, mycoplasma, and their size is smaller, generally smaller, and multiply much faster than eukaryotic cell. They may vary greatly in shape and size, but exhibit similar basic cellular organization. Now, after that, we start to discuss basic structure of prokaryotes. See, most prokaryotes normally have a cell wall surrounding cell membrane, cytoplasm present, but remember, no well-defined nucleus, naked genetic material, genomic DNA is seen. a single chromosome, inverted comma, or circular DNA. In addition to the genomic DNA, many bacteria have small circular DNA outside the genomic DNA. The small DNA are called plasmid, important term, plasmids. Episome, slightly different. Plasmid only NCERT says. Plasmid DNA confers certain unique phenotypic properties to such bacterial resistance to antibiotics. Again, plasmid DNA is also used to monitor bacterial transformation with foreign DNA. No other cell organ. Only ribosome seen. Ribosome is a membranous. After that, 5 theory question as usual and 5 NEET model question with the key. If you like this episode, I told you in YouTube I am supplying all this note, then we'll be discussing separately small segments. So it'll be convenient. Reference will be there as no. If you like this episode, subscribe or like it, share it, comment on it. Meanwhile, remember, through short, I am sending questions, preferably in the evening time. If you subscribe all, you'll be getting all this for NEET preparation. Okay. So time being, we can wind up. Thank you. Take care. Bye.